Order, and welcome to this public session of the Science, Innovation and Technology Select Committee. And first, as is our practice nowadays, we are going to welcome this week's innovator, Sebastien Ourselin. Sebastien has been brought to us by George Freeman MP, so I'm going to ask George to introduce him.
Thank you, Chair. Welcome, Sebastien. Folks, this is a very short introduction. Three things. Seb is on the front line of the medtech sector in King's over the road. And what you're about to hear is a very interesting story and a development of well, I'll let him explain it, but this is a robotics syringe. And I wanted to flag three things. Firstly, medtech, traditionally in life sciences, has been the poor relation. It's been all about drugs and therapeutics and discovery. When I entered the sector, medtech was big boxes on the top of tables. Now it's the cutting edge of diagnosis, prevention, smart health, digital health, data. Secondly, King's is often the poor relation in London. Imperial and UCL tend to get a lot of the headlines. Seb is running a major new diagnostics and medtech innovation hub at King's. And thirdly, this is completely key to the Secretary of State's agenda for reforming the NHS.
Fantastic.
Thank you very much, George. Thank you very much, ladies and gentlemen, for giving the opportunity to present today. I'm Seb. I'm Professor of HoC Engineering. I've been working at King's over the last 7 years to build what we have named the St. Thomas MedTech Hub that you can see here, which is right in front of your curtains, on the other side of the river, which is basically the largest concentration that we have in the UK of MedTech research facility embedded into an acute NHS trust, not only delivering R&D, but as well offering clinical services and transforming the life of many of our patients already on site and beyond. I want to start really to explain this through two exemplars. One which you have in your hand, which I will go into detail in a few minutes. I just wanted you just to picture in your mind how a surgical robot looks like for you. And I'm sure that if you think about surgical robotic, you will think about a very large piece of equipment, of the like of a Da Vinci on the left of this slide, or these fantastic companies in the UK, which is now the largest medtech in Europe, Cambridge Medical Robotic. Those robots have very large footprint. They are used for general surgeries, and there are clearly a very huge push right now to get more of those surgical robotic on the market and for patients, because they do benefit to the accuracy and reduce the times of hospitalizations. But what's about the other type of robots and how you will use it in different conditions? I want to just highlight hearing loss. Hearing loss is really a global crisis. We talk about over 430 million people affected. Only in the UK, 1 in 3 adults are suffering from some deaf hearing loss. Or tinnitus, and there is half of the population over 55 which have some kind of hearing loss. So this is clearly a major problem, a major crisis, and we have treatment to be able to tackle this global epidemic, but the challenge is how do you inject this treatment? One of the most common treatment for hearing loss is to be able to inject steroids in the inner ear, in the intratympanic area, and you do this manually. You do it as you see on the left-hand side of this picture here. You go to see an ENT surgeon. You must see a specialist after you've been diagnosed, and the earlier the better to be able to get treatment. Some people have been waiting for weeks to be able to see an ENT surgeon, and therefore they've got permanent hearing loss. In this condition, you need to inject the steroid, and you need to inject at the right place without creating any further comorbidity. On the right-hand side, we're talking about the revolution of gene therapy for hereditary hearing loss. So we're talking about people who had congenital hearing loss and people who actually are not able to actually hear at all, unless for some of them they will have a cochlear implant. But there is other ways, which is to inject these drugs specifically in the cochlea. And to do this, so cell treatment, you need to be very precise. Two emerging advances has been happening over the last year, and that I'm presenting here. Those two companies have created those stem cells. And the challenge is how do you demonstrate the accuracy, the efficacy? And if you have a treatment which actually is dependent of the quality of the precision of which you inject this drug, then you're going to inject noise into your clinical trial, which eventually could make it fail, and more importantly as well, could make it more expensive and difficult eventually to bring across the community at scale. So the challenge of being able to bring this technology, this drug at the right place in the inner ear or in the cochlea is really a major challenge. And if you think about robotic, you could think, well, we could bring a robot to do that. So that's how people have been looking at it. Those big robots exist. Why don't we bring them into this problem? Well, the challenge is really using a multi-million-pound solutions for a very, I will say, tiny, confined area. So what's about developing a different type of robot? This is an area of what we call endoluminorobotic. And what you have in your hand is those green bits that you could consider as a smart rock arm. And if you play with a syringe, what you will see is I can change with some of those bubbles you see in the inner ear. And by doing this, I can basically confine the syringe that I can put in it. And if you can pass this through, please.
The record, Seb, what is this we're passing around? So
we are passing around, so what you have in the green, that is small robots, these small trocars that you put in the inner ear. With those small bubbles, as you will see now on those videos, with those small bubbles, you can position exactly this trocar in the inner ear, and then you can now have a small camera, which is a black dot that you've got on front of these green bits, and then you can put your needle in it, and then you can do your injection either of steroids or eventually of your regenerative therapy. So basically, you convert a massive robotic solution into a tiny bit of the robotic solution, which is patient-specific, which can be single-use. We're talking about a few pounds to build this system, to put in the inner ear. Of course, there is all of the electronic and the AI and computer vision to add to it. But this is a very, I would say, a safe, efficient solution built in the UK, which is targeting a global market, which could be eventually available to over 430 million patients per year. Right? This is patented. This is a fantastic collaboration between two major NHS trusts, two major universities in London. It has been co-funded by UKRI, and I applaud the funding that we can get from NIHR, I4I, which I think are critical. Non-dilutive funding for companies creation is critical. This is going through a spin-out between UCL and King's as we speak, and it will be launched next year. The second example I want to present is around tumor tissue resections. So if you have a tumor, if you're unfortunate enough to have a cancer and you have a tumor, which is roughly 1 in 3 patients anyway, you will want, if it's diagnosed early, one of the most curative way of doing it, of treating it, is going to be through resection. How do you resect a tumor? Well, the challenge is you don't see sometimes the boundary of the tumor. And in fact, in 20% you have complication, and in 30% of the case, you will leave significant tumor. Leaving tumor behind means that you could have recurrence, means that you haven't treated the cancer, you just have delayed it. How do you make sure that you remove the tumor? This sentence, this claim that I'm making, are now really published widely, and it is really a big social issue in our cancer, because we know how to diagnose early now. How can we treat them efficiently through resection? We decided to focus first on colorectal surgery. This is the third most common cancer. One of the big challenges of colorectal surgery is what we call anastomotic leakage. So anastomotic is basically a complicated word just to talk about suturing. So what we need to do is to identify the part of the colon you want to resect and then stitch it. And once you have stitched it, what you hope is there is no leak. If there is a leak, then you have a major challenge, which can be deadly, and therefore you need to have an open surgery eventually to be able to correct for this leakage. This is costing over 100 million in the UK. This is costing over $1 billion in the US every year. And it's costing more than 5 extra days of hospitalization for these patients, and you've got 35% mortality rate out of this. So the way that people are doing it is to use a dye. You need to switch off the light, use an infrared camera, and you can only see it when you inject the dye. As soon as you inject it, the dye starts to dilute. What we can do is try to see how we can use white light. How can we use standard visible light, not only red, green, blue, but all of the different spectrum to try to extract new biomarker. And this is a combination of artificial intelligence, new compute available to do that in real time. The camera you see in your hand is something we have built, was funded by venture capitals over the last year. We raised over £9 million, including £6.5 million from VCs. We had fantastic support from NIHR, again, I4I. This is manufactured next door. This is UK CE mark since last year. We already had patients benefiting from this technology. In St. Thomas Hospital, we start a clinical trial with 3 sites across the UK in 3 months, and we are going for FDA approval by June. And why this is important? Look at this.
We're going to have to come to a close.
Look at this video. What you see on the left-hand side is a natural light. This is in the case of a kidney of a pig. That was a study we did with animal studies that was very important to demonstrate the value. What you see here is we clamp the main artery, and in real time, you can see the kidney becoming all purple. Why is it becoming all purple? Because there is a limited amount of oxygenation going to the tissue. So I can demonstrate to you that I can measure in real time what we call StO2, tissue perfusion, blood oxygenation in the tissue. This could be revolutionizing even the transplantations, because we don't even know sometimes when we transplant a liver if the liver is still alive. This is going to be really important in surgery right now. Thank you
very much.
I'm 9 minutes. Can I have just 1 minute more? Just
9 minutes.
I'm 9 minutes.
You said 10, did we?
Yeah.
We did, but then we started late because, one more minute.
I would like, to explain why this is important to have an infrastructure to do that. Those technology will never happen without being co-located with the hospital, without having a venture builder, accelerator, support for healthcare economics, support to go-to-market strategy. It's not only about innovation from R&D, it's innovation in the way we deliver it at scale. This happened in less than a year and a half to become a product, and it's thanks to the co-location and the infrastructure we have in place at St. Thomas with the NHS Trust, the co-partnership with industry to be able to actually build such infrastructure and as well deliver it at scale and market it.
Thank you. And thank you very much. Thank you for setting up that excellent example of partnership between the NHS research and the benefits that it can bring to the NHS. Order.